telling you what are actually entomopathogenic fungi. Well, entomopathogenic fungi are fungi that cause diseases to insects and arachnids, and also to a certain extent arthropods. And these type of organism can be found actually throughout the kingdom of fungi, but there are mainly two principal groups, which is here from the order Laburnbeniales and from the order Hypocreales. The order Laburnbeniales, they are actually ectoparasites, and they are usually like non lethal parasite. They are parasites that grow on the exterior of insects, and insects tend to be less healthy, etc., etc., etc. But this group don't usually cause lethal disease. But today I'm going to talk mainly about the entomopathogenic fungi from the order Hypocreales, because these are hemibiotrophic pathogen that cause lethal disease to insects. And because of the fact that they can cause lethal disease to insects, they are very interesting to be used as biocontrol agents against pest insects. And I will show you also today that they can be much more than that. In the order Hypocreales, actually, they are not all pathogens of insects. Actually, this order contain many species of pathogens associated to many groups of organisms. But their ancestor was inferred to be pathogens of plants, and then the entomopathogenicity has evolved multiple times in three major families called Decipitaceae, Clavicipitaceae, and Ophiocordicipitaceae. Here represent it with the red branches. But we also observe actually occasional reversion to become plant pathogen again in some group. And what is even more interesting also that we observe actually some group and some species that are mycoparasite, which mean actually they don't grow directly from the insects, but grow on fungi that are pathogens that attack insects. So we call it's a kind of mycoparasitism, like some pictures shown here. Like here, you have like a kind of like Ophiocordyceps that growing from how do we call it in English? ขออภัยแมลงสาบ cockroach, sorry. Don't sleep me. Yeah, so like you have Ophiocordyceps growing from cockroach, but then you have Pseudoneomyces, which grow as white mycelium over the Ophiocordyceps, which grow on cockroach. Yeah, it's just an example to show you that actually this group of organism is so diverse in their ecology. Sorry. And if we take an example of a major family of the insect fungi from the Hypocreales, the family of Cordicipitaceae, which is a very well-known family because of like the cordyceps, tangshao, etc., etc. We can see that although many of them are pathogen of insects, which represented here by the blue branch, but actually many of them are also can be found in the environmental samples, like in soil, etc., etc. Can infect arachnid and also nematodes. And actually there are more and more works in these recent years that show that this group of fungi can also be found associated to plant roots and rhizosphere, and can contribute to improve the growth of plants. For example, here, these are some works of some teams from China and India, which show that, for example, here, I think this is in tomato plants. Yeah. So like it's in the soil stress condition without fungi, and when you inoculate metarhizium with the seedling, it can grow better. So actually there has been like more and more this kind of work that has showcased actually the potential utility of using this group of fungi to be also a biostimulant to stimulate the growth of plants. Yeah, so actually, this group, which is basically known as entomopathogenic fungi, but actually they were shown also to stimulate the plant growth, to be able to infect nematodes, to be able to infect insects, and also produce many secondary metabolites that has been shown to have antimicrobial, including antifungal properties. So they are very interesting to also not only be used as the biocontrol agent, but also as the biostimulant in biofertilizer, or to help the plant to have a better protection against some plant disease also. So actually, and this versatility has been like more and more showcased, and particularly there is a genus that is very well known and which has been shown that they can do like so many things, not only just being the pathogen of insects, but also being associated to plant rhizosphere, contribute to growth, etc., etc. So actually, so this group of fungi is very interesting to be used in the same time as the biopesticide against pest insect, and in the same time as the biostimulant to improve the plant growth. But however, there are actually very few studies that kind of like synthesize the dual use, the use of both properties at the same time. If you go and search for literature, like there are so little overlap between when you search for the biocontrol and the plant growth. And many of the studies that exist that synthesize these two ideas are studies that are only conceptualized, but not really yet like implement it in like a big scale. So, and that's why we, so that's like one of the objective of our lab right now is like to search for a good candidate of entomopathogenic fungi that can be also used as biostimulant to improve the plant growth. And luckily we got a funding from National Research Council of Thailand since almost 2 years to look for entomopathogenic fungi that can be found in paddy fields around Thailand, and try to see whether there can be good candidates on one hand to improve the growth of rice, and on the other hand also to uh, be able to be good candidate to be used as biocontrol agents against known pests of rice, such as the brown plant hopper or the nematodes that cause root knot disease. So we try to find good candidate at the laboratory scale, implement at the greenhouse scale, and if we have good results, well, we would love to implement in the field scale also. So actually, we have sample across Thailand soil samples from paddy field and isolate them. Like the protocol is not like very complicated. It's just kind of like dilution of soil with sterile water and incubation on selective media, and then we exclude all the fungi that are known like contaminants and known fungi that can be found in environments such as like aspergillus, etc., because we are mainly interested in the fungi that have potential entomopathogenicity, and we identify on based on the morphology and the ITS barcoding. And so all around Thailand we found 75 strains of soil-borne fungi from paddy fields, and the majority of them were identified to belong to the Purpureoxynium and Mithracium genera, and some from the Pogonia. And actually, Purpureoxynium is a genus of the Hypocreales that is already well known to be soil-borne and actually have the pathogenicity against nematodes. There have been like many works that have published about that. And then Methylrhizium, well, as we know, like right now, more and more also showcased to be soil-borne and contribute to the plant growth. And so actually we select representative strains from these genera to search for good candidates that could be used as biostimulants. Like a microbe can contribute to the plant better growth of plants in so many ways. It can help plants absorb better the nutrients. It can solubilize nutrients in soil and make them more available to the plants, or they can also like produce analogs of growth hormones, plant growth hormones that can help plants grow better. So like we start by looking for those that can solubilize well the phosphate, because the assays to look for phosphate solubilizing strains are not very complicated. You have a special media called Pikovskaya that you can use to inoculate fungi on it, and then the phosphate solubilizing ability is just measured as the fungi produce acids that will solubilize calcium phosphate, which is the only source of phosphate in this media. It will produce halozones around the colony. So like the proportion of the halozone compared to the size of colony will give you the phosphate solubilizing index that give you like the idea whether it can solubilize well or not the phosphate. And here you can see that Metarycium in general tend to solubilize phosphate way much better than Purpureochilium and Poconia. So apparently, like Metarycium is also a very good candidate as a plant biostimulant. So actually, we picked randomly two of the best strains, the MY 12317.02 and 0.32, to try to make some tests at the greenhouse scale. So like we grow rice in sterilized autoclave soil and inoculate our candidate strains when we grow them, compared to the control where we don't inoculate anything. And then at different stages of the growth of rice, we measure their phenotypes at the seeding stage, the vegetative stage, and the budding stage. And then also at the maturity, we harvest the panicles to measure like the size, the proportion of developed kernel, etc., to see whether it contribute also like to better yield of the rice. And the phenotypes have been evaluated at the NASDA high throughput phenomics facility. So basically, like you have camera around the plant with sensor that will take pictures from top, from bottom, and from the picture, it will calculate and give you the digital volume of the plant and the height. And we also have sensors that can measure the photosynthetic efficiencies. And so here are the results of our first year. We can see that in terms of the digital volume of the plant, particularly at the vegetative stage, like, oh, I forgot to tell you, we did experiment in two conditions, the wet and dry condition. Wet condition mean like we water the plants abundantly so that there will be like stagnant water in the pot, just like in the rice field. But the dry condition is just like you don't water the rice every day, just every 3 to 4 days, and the soil will never get stagnant water in it, because like you have like different kind of rice culture, right? Cultivation. And sorry. Yeah, so here, particularly at the vegetative stage, with the wet condition, we can see that the treatment which received the two fungal strains can grow majorly better than the control treatment that did not receive the fungal strains. And but in terms of the height, for the wet condition is not different, but for the dry condition, yes. And also like this at the seeding stage, there are some like difference that maybe not all significant, but a tendency to higher plant digital for the dry condition with the strain 32. And at the booting stage for the dry condition also, the strain 32 give a higher plant digital volume and higher plant also. So actually we have like phenotypes that are shown to be better when the plant receive the fungal candidate strains compared to control. In terms of photosynthetic efficiency, you can see here that, for example, for the wet condition, like the control and the treatments, they have like equivalent potential photosynthetic efficiency because like the plants are abundantly water and do not have any like hydric stress. But in the dry condition, like their photosynthetic efficiency is highly reduced in the control. But when we add the fungal candidate strains, like the photosynthetic efficiency is also increased, and particularly it became significant at the booting stage, particularly in the dry condition. And in terms of the yield and the production of the panicles, we can see that the strain MY.02 give actually longer panicle in the wet condition compared to control, higher dry weight, and also more developed kernels or like good kernels. So actually, but in the dry condition is no difference. So we can see that like our fungal strains can contribute to better phenotype in various aspects of the growth and also the reproduction. And so we want to know whether our two candidate strains, the MY.02 and .32, can also be used as biocontrol agents against brown plant hoppers, which is a major pest insect to rice cultivation. So like we just test them at the laboratory scale. So like you just like spray the fungal suspension at a controlled concentration on the brown plant hoppers, and as you can see here, both strains result in mild to high mortality of the brown plant hoppers compared to the control where we sprayed blank spore solution. So actually we have candidate strains that could be interesting candidates as biostimulant, biofertilizers, and also biocontrol agents. We also test our strains isolated from paddy fields and also other strains from our culture collections on nematodes that cause root knot disease. So basically, like you incubate eggs or juveniles of the nematodes in spore suspension or the culture filtrates for 72 hours and then take out those eggs and juveniles to inoculate on rice seedlings and let the rice seedlings grow for a few days, take them out, do specific coloration to be able to visualize the nematodes, and you can quantify the virulence of the nematodes. And actually, we see that, I don't know why it appears smaller than on my computer. I'm sorry. Here, the control, this is the virulence of the nematodes. In the control treatment where there was no fungal inoculation, like the virulence of the nematodes is 100%. But if we inoculate the entomopathogenic fungi from various genera, like the virulence of the nematodes is notably reduced. And we have like a very interesting candidate here that actually can reduce like up to 80% the virulence of the nematodes in the root to the root invasion in rice. And here we take that candidate strain to make more tests at greenhouse scale by co-inoculating it with nematodes eggs, and then to let them go like for 2 months, and then evaluate the severity of the root knot disease by counting the number of knots formed by the nematodes on the right on the roots. And we can see that here, this is like the control where it did not receive any fungal inoculation. And like, so you can have like more than 300 knots formed by root knot nematodes. But when we apply the candidate strain, whether using the filtrate or the spore suspension, so the severity of the root knot caused by the nematodes are significantly reduced, and particularly when we use the spore suspension in blue, more than only using the fungal or the culture filtrates. So basically, to summarize this work, entomopathogenic fungi can be found throughout the kingdom of fungi, but only the entomopathogenic fungi from the Hypocreales are hemibiotrophic pathogens to insects, which have been now demonstrated to be much more than pathogen to insects. It can be found in soil associated to plants rhizosphere, can infect nematodes. So from our screening, we found like good strains that can be good candidates at biostimulants in biofertilizer for rice cultivation, and also good candidate as biocontrol agents against brown plant hoppers and the root knot nematodes. So actually there are a lot of variation between strain and species that warrant further research. And I just want to emphasize on something, because apparently like today we are in a workshop about the platform on proteins and biochemicals of fungi. What we have been doing in our lab is nothing of like advanced technological platform or anything. Actually, most of the work in trying to use fungi as candidate strains in biofertilizer or biocontrol agents, the screening has been always kind of blindfold. Like you have a big collection, a lot of collection, but you have no choice but like picking them up randomly and try to test them with some knowledge of the taxonomic group, etc., etc. You can better target, because you know that, okay, Metheraceum and Bovaria wouldn't be like more probable to be able to infect insects than like other species or genera that have been found to be really specific to some insects. But basically, when you develop this kind of research, it requires actually a lot of manual work and determination to go through. Yeah, and sorry. I would like also to emphasize the fact that why it is important to screen a large collection. For example, here, this is some preliminary results of our test on brown plant hoppers. But this time we took brown plant hoppers from 3 different populations from Phetchabun, Pathalung, and Buriram, and test whether different strains of entomopathogen. I'm sorry, I'm a little bit high today, like whether like different strains of entomopathogenic fungi can be like equivalently virulent against different populations. And we can see that, for example, like the same strain do not always give the same result with different populations of the brown plant hoppers. For example, the BCC 4849, which is actually a champion candidate strain that is used by Biotech in our biocontrol program. It is very virulent, like around 80 or more than 80% virulence against the brown plant hopper from Pathalung and Buriram. Why in from Phetchabun, it's only give pretty like average entomopathogenicity. But another strain is more interesting for the brown plant hoppers from the Phetchabun population. So actually, that may be like because here we are doing research about also in the same time, like we want to do application on biocontrol agent, biofertilizer, but in the same time, this group of organism are pathogens of insect, and they are kind of post-pathogen interactions in the details that we don't understand yet for the whole picture. So what we would like to do also is to actually screen our collection and link the genomic data to the virulence data and try to understand why some strains are more virulent than others, whether they could be like related to the presence absence of some secondary metabolite gene cluster that are known to produce mycotoxins, whether they can produce some kind of like virulence factors, etc., etc. So this is a kind of like a fundamental research aspect that we are going further right now. So all this work would not have been possible without so many people and so many helping hands. First of all, from my lab, led by Dr. Jennifer Leung Saat. So actually, we are a research team which does not really like have much experience on the plant physiology or the application research in the biocontrol or agriculture. We have been researching the taxonomy and biodiversity of fungi like for the last 20 years. But right now, like nobody want to give you money to do only taxonomy and biodiversity research, right? So if you want to survive, you have to adapt. So, but we know that like we have such valuable resource in our collection, and that our strains could be like really interesting to get more attention and application in the future. And I would like also to thank Dr. Chintana Anapngam and Dr. Pontip Ryungpanan from Kasetsart University, Kamphaeng San campus, because this project on rice has been part of a big project led by Dr. Chintana, and Dr. Pontip is the one who managed the assays on the root knot on the nematodes causing root knot disease. And I would like to thank Dr. Catherine and Surian from the Innovative Plant Genetic and Physiology Research Team at Biotech, who have taken care of the phenomics and the phenotyping of our plants, and also Dr. Alongkorn for his valuable advice on various aspects of the biocontrol of entomopathogenic fungi. So thank you very much for your attention. And if you would like to discuss more, you can contact me with this contact detail. You can go to see the website about like the research developing by our team, and also Twitter if you would like to follow. Thank you very much.
Thank you, Dr. Noppol, for your enlightening presentation. We have only 1 minute for very quick questions. Yeah.
Thank you so much for your presentation. I can relate to what you say about biodiversity work not being that sexy for funders, and I've experienced the same thing too in Indonesia. One thing that I'm curious about, one issue with working with entomopathogenic fungi when it comes to application is that they require a long time to grow, right? So have you ever looked into the diversity and see if there's any specific species that can grow faster, or perhaps have there been any efforts so far to speed up their growth, perhaps?
Yeah, sure. Metarhizium and Borealia, which are like two major genera that are currently used in biocontrol programs in various areas around the world. These two genera grow quite fast. They can grow quite easily, like at the laboratory, on PDA, on social media. They are not very slow, like Borealia and Metarhizium. From the isolation to like a full plate of culture, it can take just a few weeks. But there are like other genera, other species, many species from Ophiocordyceps are very fastidious, and they are also like very specific to like certain group of insects. And that's why maybe like they are also like very fastidious, because there must be some kind of very specific signals from insect hosts for them to grow. So like for those group, they are not interesting to be candidates for the biocontrol, because like too slow to grow and then too specific to some group of insects. But if you want to use entomopathogenic fungi in biocontrol or biofertilizers, like well-known groups such as Bovaria or Metarhizium shouldn't be okay. Yeah, yeah.
Thank you, Dr. Noppol.
Thank you.